This is cell. Whenever stress will come, human cell first will do adaptation, 5 different type of adaptation. Second, it will do injury. It may be reversible injury. It follow irreversible or cell death. That irreversible injury is also called as cell death. There are two types of cell death, apoptosis and necrosis. Now we'll discuss about apoptosis. Apoptosis is a type of cell death in which a series of molecular stuff in a cell lead to death. How cell select whether it want to die either apoptosis or necrosis, whether it want to die either with apoptosis or necrosis. Why two type of cell death? So how we will define? It is a cell suicide. Apoptosis is a cell suicide. Why cell want to do suicide? There are two different type of reason. Either cells in human body already performed function, no more use. Otherwise, the cell DNA of the cell is damaged to such a extent it cannot be beyond repair. Now, this can suicide, otherwise can converted into cancer cell. So, according to Robin, it is a type of pathway. It is a pathway of cell death that is induced by a tightly regulated intracellular program in which cell destined to die activate enzyme caspase enzyme, degrade the cell own nuclear DNA and cytoplasmic protein. It is a pathway of cell death that is induced by tightly regulated intracellular program in which activate enzyme caspase, degrade the cell own nuclear DNA and cytoplasmic protein. Here there is no leakage and no inflammation. So there is no leakage outside and there is no inflammation. It is the way elimination of unwanted cell and those cell which are damaged beyond repair capacity of that cell. If the cell want to commit suicide, either already performed or DNA damaged. So in that cytoplasm of the cell, there is an enzyme known as caspase enzyme. Inside presented enzyme caspase enzyme. All the caspase enzyme are inactivated normally. All human cell have that caspase enzyme in that cytoplasm, but there are inactivated. When the cell received the signal that it has to commit suicide, when signal received by the cell, okay, after that only that caspase enzyme become activated. All the human cell having caspase enzyme inactivated form. When that cell damaged or already performed, when it have that signal, after that only that caspase enzyme activated. Caspase 1, 2, 3, many are become activated. So many enzyme is there, they become activated. After become activated, they will split entire cell into multiple small apoptotic bodies. So after activation, they will converted into small apoptotic bodies. Apoptotic bodies containing small portion of cytoplasm, small portion of cell membrane, and a small portion of nucleus. So it containing membrane, nucleus, all the things, entire cells. Okay, then cell membrane and nucleus, cell membrane and nucleus, everything splitted into small multiple portion, which is known as apoptotic bodies. This name is called as apoptotic bodies. During this phenomenon, no leakage outside, there is no inflammation. No leakage and no inflammation outside. Necrosis, leakage and inflammation present, not in apoptosis. Necrosis have leakage and inflammation, but apoptosis no leakage and no inflammation. So apoptotic bodies are formed ultimately. A cell is known as, now this one is the phagocyte cells. Okay, is coming and phagocytes in helping all the apoptotic bodies. This phagocytic cell will eat all apoptotic bodies, so no leakage and inflammation is absent. Actually, apoptosis generally involved in a single cell. It is a group of cell. It affected only single cells. Single cells in contrast to necrosis, but usually involved in group of cell. Necrosis means group of cell. Apoptosis means single cells only. Next, types of apoptosis. There are two types. One is physiological, second one is pathological. Physiological, programmed cell death. Pathological, unprogrammed cell death. Physiological, best example, embryogenesis. What is embryogenesis? This is the uterus of mother inside fetus. Fetus forming inside of that uterus of mother. So during this formation, embryogenesis in various organs are formed. For example, here hand, this one is the hand initially, like this only. Initially block, here a block only, no digit. So later on 5 digits is formed. How it formed? The cell between the digit are died. So in between the digit, okay, the cell are died. They are doing apoptosis by using apoptosis mechanism, that in between that cell are died. After that only 5 digit fingers are formed. Not only this, multiple organ formation, this apoptosis really helpful. So this is best example of physiological causes of apoptosis. Second example, elimination of potentially self-reacting lymphocytes. What is lymphocytes? It is a type of WBC which is killing foreign cells. Sometimes these lymphocytes in human body, they become abnormal, they become self-reactive, they become autoimmune, they kill body own cells. So these lymph nodes which become abnormal, they eliminated from the body. This is apoptosis. It is a physiological phenomenon. So first one is embryogenesis, second one is elimination of potentially self-reacting lymphocytes. Second pathological condition, cell death in tumors exposed to chemotherapeutic agent. Then progressive depletion of CD4 T cell in that pathogenesis of AIDS. Next, cell death in viral infection. For example, formation of Councilman bodies in viral hepatitis. Next, mechanism. There are two types of mechanism in apoptosis. First one is extrinsic factors. extrinsic pathway and intrinsic pathway. This one is the cell. So this one is the nucleus of that cell. This one is the DNA of the cell. DNA of the cell. The cells has to undergo apoptosis. Now, whether the signal coming from that outside, the signal coming from outside of that cell name is called as extrinsic factor. Coming from inside of that cell name is called as intrinsic factor. Both are activate caspase enzyme by two mechanism, intrinsic and extrinsic pathway. Extrinsic pathway, it is initiated by extracellular stimulus which help of specific receptor called death receptor. Intrinsic pathway, it is a result of increased mitochondrial membrane permeability and released pro-apoptotic marker like cytochrome C into that cytoplasm. Now mechanism. Each of these two pathways have two phases, extrinsic and intrinsic. Extrinsic having two phases, initiation and execution. Like that, intrinsic pathway also having initiation and execution. These both initiation are different, both executions are same. This one will activate caspase enzyme 8. This one will activate caspase enzyme 9. Initiation phase of extrinsic pathway, it is initiated through specific receptor called two type of receptor, FOS protein CD95 and TNF receptor, tumor necrotic factor. So see the cell. Any cells of that body have receptor on their surface. Okay, all the cells having receptor. These are known as death receptor. This receptor name is called as death receptor. There are two set of death receptor, 1 and 2. Either of two FAS receptor or this TNF, tumor necrosis factor receptor, are present. So the cell is surrounded by death receptor. So this is the cell. It is surrounded by either 1 or 2 receptor. What are the 2 receptor? FAS, FA protein receptor, then tumor necrosis factor receptor, 2 receptor is present. So this death receptor present distant, okay, distant from each other. Now ligand. Now this ligand is coming, it bind with receptor. It is signal. It is coming from outside. That's why it is called as extrinsic initiation pathway. So once that ligand coming and binding, multiple FAS, that receptor FA proteins or that receptor come closer. So here distance, here it come closer. Once the ligand coming, once it is binding with the FAS receptor, multiple FAS receptor come closer, they become activated. They form domain. Okay, they form domain. So they are known as FADD, fast associated death domain. So all come together, so they form domain. That domain name is called as FAS, associated death domain. Once that FADD is formed in that cytoplasm, of that cell, various caspases are presented. We know 1, 2, 3 and so on, but all of them are inactivated. This FADD will activate caspase enzyme 8. So many enzyme is presented. 1, 2, 3, 4 like that, but it will activate caspase enzyme 8. From inactive state it become active state. Now that caspase enzyme 8 activated. Initiation phase of intrinsic pathway, signal will come from inside. The intrinsic signaling pathway that initiate apoptosis involved a intracellular signal. Inside of the cell signal will get, okay, intracellular signal within the cells or mitochondria initiate event between the two layer of mitochondria. Between two layer of mitochondria there is a protein. Inside protein is presented. Cytochrome C never come out in that cytoplasm. So inside it will never come out from that cytoplasm. It is presented in between two layer of mitochondria. Once it will come outside in that cytoplasm, it causes apoptosis. Once it will come to outside, it causes apoptosis. This is the cell. This is the mitochondria. This is nucleus and DNA. Mitochondria transit. Okay, here this one is the inner layer, this one is the outer layer of mitochondria. Now mitochondria have a gate. Gate is known as transit. Okay, gate name is called as transit. To who is preventing that cytochrome C. It will prevent that cytochrome C to come out of that transit. Cytochrome C not coming out of transit. So because here gate is presented, that gate is name is called as transit. So it never come from outside. Goose who's preventing here two squad is presented. That squad name is called as BCL2 and BCLX. These are preventing cytochrome C to come out. Both are preventing not come out, that liquid not come out, because here transit is presented, two type of transit, that name is called PCL2 and PCLX. These two protein are known as apoptotic protein. Both are having that name, anti-apoptotic protein, because they preventing apoptosis. Still DNA is okay. Now DNA got damaged. So now this DNA got damaged means DNA sends signal to mitochondria. So just give that cell. So it become damaged. So the cell cannot survive. That cell not survive, now come out into suicide, we have to die. So the signal come from inside, not coming from outside. So it is known as intrinsic pathway. So once mitochondria receive that message, a signal from DNA, the two squad, we know these two squad replaced by three new squad. What are the three new squad? This POX, POP and PEM. POX and PEM. These are pro-apoptotic squad. They will open the door. They will allow cytochrome C to come out. So this two squad name is called as anti-apoptotic protein. This three squad name is called as pro-apoptotic protein or squad. So now it allows cytochrome C come out. So what will do? It will activate caspase enzyme 9. It will activate caspase enzyme 9. So pro-apoptotic caspase 9 is converted into active caspase 9. So the two squad replaced new squad. will allow that cytochrome leakage, so increase the mitochondrial permeability, so release the cytochrome C. After that, activate caspase enzyme 9. So, extrinsic factors pathway activate enzyme caspase enzyme 8, intrinsic pathway activate caspase enzyme 9. So both are activated. So these two enzyme will activate caspase enzyme 3 and 7. After that, it will activate other caspase enzyme also. So caspase here all the enzyme will activated. After that, it causes cell degradation or apoptotic bodies. That apoptotic bodies formation, then phagocytic will come, it will help that apoptotic bodies. This one is the okay. So cell enhancement process will happen. Now phagocytes will come and eat that apoptotic cells. How phagocytes recognize which one is the normal cell, which one is apoptotic cells, what is the mechanism? So now phagocytes recognize cell which on that phosphatidylserine. Phosphatidylserine normal cell means which is presented inside. Okay, normal cells have protein. That protein name is called as phosphatidylserine. That protein is presented inner surface of that cell membrane. Okay, during apoptosis there is flip-flop ulta. Okay, flip-flop because of that flip-flop like this presented. Okay, this phosphorus detail serine is coming on that surface. Okay, it will come on that surface during apoptosis. So only phosphatidylserine presented on that outer surface. Okay, presented on outer surface. That phosphatidylserine presented means phagocytes not engulfed. Presented inside means not engulfed. So phosphatidylserine is acting as a marker. So it acts as a marker for phagocytes it eat. So phosphatidylserine presented on outer surface. Outer surface phosphatidylserine presented means here enhancement process will started. It acts as a marker for phagocytes and it eat outer surface, so it will eat. Next one is what are the morphological changes in apoptosis? So this one is the it having 7 future. This one is the normal cells. Here normal cell it committed suicide means next second stage it become more pink because of shrinkage. The cell shrinkage here. Mitochondria, ribosome, endoplasmic reticulum come closer. So here all the things will come closer. So because of that shrinkage will happen, they come closer in that cytoplasm here more pink in color. So it is called as eosinophilia. This condition name is called as more pink in color. So this condition name is called as eosinophilia. Shrinkage and eosinophilia. Next one is pycnosis of nucleus. So undergo this one pycnosis of nucleus will happen. So condense. After that fragment. That nucleus fragmentation of nucleus. Here single nucleus, here fragmentation. So after that it leads to that karyotherix. It is known as karyotherix. Cell membrane formations. Okay, so here that cell membrane, that formation, Bebel's body formation. Fifth future, Bebel's body formation. Then formation of apoptotic bodies, small bodies. After that phagocytes will come and eat. These are the seven morphological changes. First one is normal cell, then shrinkage because mitochondria, ribosome, endoplasmic reticulum come closer, so it pink color, so it is called name eosinophilia. After that pycnosis of that nucleus, after that fragment of nucleus, then race formation, that nucleus fragmentation, that process name is called as karyolerics. After that, that fibril formation, then formation of apoptotic bodies. After that, phagocytes will come and eat that cell. So stringage, pink color, eosinophilia, condensation, that fragmentation of nucleus, karyolerics, then Pfeffel formation, then formation of apoptotic bodies, then phagocytes will come and eat. Then next one is diagnosis of apoptosis. How you will do diagnosis of apoptosis? There are two ways. First one is marker. There is one marker. We are using that marker, name is called as anosine 5. Anosine 5. This phosphacetylserine is present on that inner surface membrane of the normal cells, but it is flipped out on that surface during apoptosis, so it acts as a marker. This anosine 5 acts as a marker. By using this marker anosine 5, it is available in that form of liquid. Okay, it is available in the form of liquid and spread over that slide. Only apoptotic cell will take anosine 5. Others will not take anosine 5. Wash the slide after washing. Apoptotic brown in color. So brown in color. Normal cell presented like this. Apoptotic cell presented like this. Other cells normal color. This apoptotic cell is brown in color because this anosine 5 acts as a marker. Why apoptosis cells will take this marker, other not? Because it is presented that phosphatidylserine presented on that surface. Second method, agarose gel electrophoresis method. Agarose gel electrophoresis method. Normal cells. This one is the normal cell, this one is the apoptotic cell. Normal cell, single piece of chromosome, so single piece of chromosome. So this one multiple piece of chromosome, 1 2 like this multiple. It look like a step ladder appearance. So by using these two methods, we can identify which one is the normal cell and which one is the apoptotic cell. So in this video discussed cell apoptosis. Apoptosis takes place in single cell. Necrosis, group of cell. Here cell shrinkage will happen, but their cell swelling will happen. Here pycnosis and karyolerics takes place. Their karyolysis will takes place. Here plug formation, no leakage and no inflammation, but there leakage, inflammation will happen. Here main enzyme, caspase enzyme, necrosis hydrolytic enzyme. So here caspase enzyme 8 9 2 3 7, all the enzyme activated, that hydrolytic enzyme in necrosis. So these are the major difference between necrosis and apoptosis.